Hello, my name is Dr. David Seiden, and I'm going to be reviewing anatomy and some related topics with you. I'm a professor in the Department of Neuroscience and Cell Biology at the Robert Wood Johnson Medical School at Rutgers University in New Jersey. I've been on the faculty there for about 45 years, and I've been doing programs like this for Kaplan for about 25 years. So what we will be doing is reviewing material that is high yield, the material that's most important for you to know in preparation for the USMLE. This will be, as you probably are aware, a very clinically oriented approach because the questions that you will see on the USMLE are primarily in clinical vignette format. So we want to focus on that material that is most likely to get incorporated into a clinical vignette, which means the clinically oriented anatomy. So what we will be covering is gross anatomy. We'll also cover embryology and some histology and a little bit of radiographic anatomy. Now, the way we will cover these additional topics, firstly with regard to embryology. Embryology is learned best by learning it along with the adult gross anatomy. And that way we can use the embryology to help us understand the adult anatomy and to understand what are the errors in development that lead to congenital defects. So what we will be doing for embryology is we will start right from the beginning doing some early embryology. We'll start from the formation of gametes and go through fertilization and early development up until about 4 weeks of development. At 4 weeks, the embryo has assumed the adult body plan, and so once we get to that stage, we will leave the embryo and then move on and do adult gross anatomy. Each time we get to a different organ system in adult anatomy, we will go back to that 4-week-old embryo and take some small portion of it and develop it to the adult to develop that particular organ system. So, for example, when we do the anatomy of the respiratory system, that's when we'll do the embryology of the respiratory system. When we do the anatomy of the cardiovascular system, that's when we'll do the embryology of the cardiovascular system, and so on through the body. With regard to histology, we will do a little bit of some generic histology, particularly with regard to epithelial cells, and we'll do that early on in the program. We'll go over the basic cell biology of epithelial cells because it's the epithelial cell in most organs that are really the business cells, the cells responsible for the primary function of those organs. So we want to pay particular attention to epithelial cells. And then after doing that, we will again do the histology of particular organ systems at the same time that we do the gross anatomy of the organ system. So again, when we do the respiratory system, the anatomy of the respiratory system, we will do the histology of the respiratory system. When we do the anatomy of the GI tract, we'll do the histology of the GI tract, and so forth through the body. And lastly, with regard to radiographic anatomy, we want to pay particular attention to the interpretation of CT scans. The presentation of CT scans and MRIs is always found on the USMLE, and so we want to make sure that you are feeling comfortable in reading a normal CT. So the way we will do that is when we complete a major section of anatomy, for example, when we complete the thorax, we'll then review the thorax, the material that we've covered. We'll review it using CT scans as a way of reviewing the material and also, at the same time, getting some practice in reading CTs. And then we'll do exactly the same thing, for example, when we finish the abdomen. We'll review all of the abdomen using CTs. Okay, so that's our plan. So let's proceed. We will start off with early embryology. Let's begin then with early embryology. So our objectives in this section will be to define the stages and timelines of embryonic development from fertilization through 4 weeks of development. We'll want to describe how the gonads develop and what happens in the gonads in terms of meiosis, specifically for spermatogenesis and oogenesis, and develop the three-layered embryo and see what is derived from the basic germ layers, the ectoderm, the mesoderm, and the endoderm. So how we will proceed is we will start off with gametogenesis, how we form gametes, how those gametes fertilize, the cell divisions that occur during the first few days after fertilization, called cleavage division, implantation at the end of the first week, and then we'll see how the embryo forms as a two-layered embryo and how that converts to a three-layered embryo, and then how that three-layered embryo folds into the adult body form, body folding. And those events correspond to the first 4 weeks in this way. From fertilization through implantation is what happens in the first week. In the second week, the embryo assumes a two-layered form. And then in the third week, it converts to a three-layered embryo. And then in the fourth week is when the body folding occurs. So we'll start off taking a look at how we form gonads. Now, on this image, what we're looking at is a schematic of a early embryo of about 5 weeks of age or so, but the specific age of the embryo is not the important point. What we want to call attention to here are these two long ridges of mesoderm on the right and left side of the back of the abdomen. Those ridges of mesoderm are called the urogenital ridges. Urogenital ridges. And the urogenital ridge has two parts. A larger, much longer, more lateral portion that's shown in the lighter shading here, and that's called the nephrogenic portion of the urogenital ridge. Nephrogenic meaning that it's going to be related to the formation of kidney structures. We're not going to talk about that right now. We'll come back and talk about the nephrogenic portion later on when we develop the kidneys, when we're doing the urinary system. The other part of the urogenital ridge, this smaller, more medial portion, is called the gonadal ridge, or the gonadal portion of the urogenital ridge. And that's the mesoderm that will form a gonad on the right and left sides. Now, even though this is where the gonad is going to form, the cells that are going to give rise to the gametes actually don't arise here. They arise outside the embryo in the wall of the yolk sac. So they arise here in the wall of the yolk sac, and then they have to migrate from the yolk sac through the umbilical cord into the embryo, to the back of the abdomen, and then into the gonadal portion of the urogenital ridge, where they will infiltrate the mesoderm and then start living in that mesoderm. And then they will go on to develop into gametes, and we'll talk about how that happens in just a few moments. But in addition to the fact that they will go on to become the gametes, those cells have another important role, and that is that their function is to induce this mesoderm to become a gonad. In other words, even though we're saying that this mesoderm is destined to become a gonad, it will not become a gonad unless it's induced to do so by the invasion of these primordial germ cells. And so if these primordial germ cells don't reach the gonadal ridge and don't infiltrate the gonadal ridge, then that induction will not occur and a gonad will not develop. And this mesoderm will remain undifferentiated and eventually will degenerate, and there'll be a missing gonad. Okay, so the absence of a gonad is typically the result of a failure of induction by these primordial germ cells because they fail to infiltrate the gonadal ridge. But, as I said, normally they do infiltrate the gonadal ridge and they will induce the formation of a gonad. So that brings us to the question of what kind of a gonad will it be? Is it going to be a testis or an ovary? Or to make that a larger question, we will say, is this embryo going to have a male or a female genital system? Now, we know that the gender or the sex of the embryo is going to be determined at the time of fertilization. And if this fertilized embryo is an XX embryo, in other words, if it's fertilized by a sperm that has an X chromosome, then this will be female. And if it's an XY embryo, in other words, if it's fertilized by a sperm that contains a Y chromosome, then this would be male. An important point to keep in mind is that the reason a male is because he has a Y chromosome. And the reason a female is because she does not have a Y chromosome. In other words, a female is not female because she has two X's. She's female because she doesn't have a Y. Another way of saying that is that the embryo is predestined or pre-programmed to become female and will become female unless something is done to force it to become male. And what forces it to become male has something to do with the Y chromosome. So if there is a Y chromosome, it will become male. But if there isn't a Y chromosome, then it will become female as it was pre-programmed to do. So that leads to the question of what is it about the Y chromosome that accomplishes this? There is a region on the Y chromosome called the SRY segment of the Y chromosome, SRY standing for sex-determining region of the Y chromosome. And the SRY segment codes for something called testis-determining factor, which we can abbreviate TDF. And so TDF, if there is a Y chromosome, we'll have TDF, and TDF will cause that gonad to become a testis. So in the presence of TDF, the gonad will become a testis. But if in the absence of TDF, in other words, if there is no Y chromosome, therefore there is no TDF, then that gonad will become an ovary as it was pre-programmed to do. So that's how we'll determine whether there'll be a testis or an ovary, by whether or not there is testis determining factor. But of course, there's more to being a normal male than just having a testis. We need to have all of the other components of the male genital system, the ductus deferens, the epididymis, the prostate, and so forth. So how do we get all of that? Well, once we have a testis, the endocrine cells of the testis, the Leydig cells of the testis, secrete testosterone. And during embryonic development, there is a period of very high production of testosterone. And in the presence of testosterone, as well as a derivative of testosterone called dihydrotestosterone, in the presence of those two hormones, all of the remainder of the male genital system is developed. So the ductus deferens, the prostate, the seminal vesicles, the penis, the scrotum, and so on and so forth, all of those components of the male genital system develop as a result of the presence of high levels of testosterone coming from the testis, of course, being caused by the presence of TDF. Now, remember that we said that this embryo was pre-programmed to become female, which means it's pre-programmed to develop a female genital system. In other words, it's pre-programmed to have uterine tubes and a uterus and a vagina. So if you want this to be a normal male, not only do you have to form the male genital system, you have to prevent the formation of the female genital system. And the way that is accomplished is with a substance called Mullerian inhibiting factor or Mullerian inhibiting substance, being abbreviated here MIF, Mullerian inhibiting factor. Mullerian refers to the Mullerian duct, another name for the paramesonephric duct, which is the embryonic duct that gives rise to the female genital tract, uterine tubes, uterus, and so forth. So if we inhibit the development of the Mullerian duct, we'll inhibit the formation of the female genital tract. Mullerian inhibiting factor comes from the Sertoli cells, another cell type found in the testis. So again, we have a testis because of the TDF. We've already said the testis will give us the testosterone, and now we're saying the testis gives us Mullerian inhibiting factor, which will inhibit the formation of the female genital tract. So with all of those factors acting together, we end up with a testis and with a normal male genital system and without a female genital system. But if there is no Y chromosome, so there is no TDF, then instead of a testis, we have an ovary. And with an ovary, we don't have the high levels of testosterone, so we don't develop a male genital system, and we don't inhibit the Mullerian duct, so we do have a female genital system. So there are no factors necessary to give us a normal female with an ovary and a female genital system. Okay, so that's how we determine whether it's male or female. If you think through the possible abnormalities here, you can see how you could end up with embryos that have mixed gender abnormalities. For example, if we had an embryo that had a Y chromosome and therefore it had TDF and therefore it had a testis and was secreting testosterone and dihydrotestosterone, but let's say for whatever reason it wasn't producing Mullerian inhibiting factor, or it wasn't producing the receptors for Mullerian inhibiting factor. Well, then that embryo would have a testis, would have a male genital tract, but would also have a female genital tract. So that would be one version of a mixed gender abnormality because of abnormalities of this grouping of normal factors. Okay, so now we have a gonad, and so we have to see how that gonad is going to give us gametes. Now, the production of gametes, or gametogenesis, occurs through a process of cell division called meiosis. Meiosis is a two-cell division process. Therefore, we have what's called meiosis 1 and then meiosis 2. This is in contrast to the cell division of somatic cells, which is by way of mitosis. So all of the cells of the body, except for cells involved in gametogenesis, all the cells of the body that divide by mitosis. But we develop gametes by the cell division process of meiosis 1 and meiosis 2. Now, this meiotic division is represented on this slide here. And what I want to do is go through this slide three times. The first time we go through it, we'll talk about the things that occur that are the same in male and female gametogenesis. In other words, in both spermatogenesis and oogenesis, what happens that's the same. And what we will see is that what's happening that is the same is what's happening in the nucleus of these cells. So as we discuss that, each of these circles will represent the nucleus of those cells. The second time we go through it, I want to talk about the things that are different in males and females. And we will see that what is different is what's happening in the cytoplasm of these cells. So when we talk about that, we'll have the circles represent not the nucleus, but the entire cell. And then the third time we talk about this, I want to talk about the timing of these events. When do the events that we will have just discussed, when do they occur? Because we will see that that is different in males and in females. So let's start with talking about what happens that's the same in spermatogenesis and oogenesis, the same in males and females. That's what's happening in the nucleus. So we start off with the nucleus in these cells here. These are the cells that basically are the daughter cells of those cells that migrated from the yolk sac into the gonad and have been living there in the gonad. These cells are going to be called spermatogonia or oogonia, depending upon whether it's male or female. But the important point to note is that these are somatic cells. These are not gametes. These are somatic cells, which means they divide by mitosis. And so when they divide, they just produce more of themselves. Then at some point, and we'll talk about when that point is a little later, but at some point, some of those cells will differentiate into cells that are no longer dividing by mitosis, but rather will start dividing by meiosis. They will differentiate and become cells that are meiotically active, and therefore they enter the meiotic cycle. Once they do that, we change their name, and instead of calling them spermatogonia or oogonia, we now will call them primary spermatocytes or primary oocytes, depending upon whether it's male or female. Okay, so that's what we're talking about with this cell here. And this cell, this primary spermatocyte or primary oocyte, will start to go through the first meiotic division. Now, the first event that has to occur in cell division, whether it's mitotic or meiotic division, the first thing that has to occur is we have to replicate the DNA. We have to double the amount of DNA that's in the nucleus through DNA synthesis. And so we have a replication of the DNA. And what's represented here to illustrate that is the following. What we're seeing in this cell is one pair of chromosomes. One pair of chromosomes. Remember, there are 23 pair of chromosomes, so this would represent one of those 23 pair. So there's a total of 46 chromosomes, and that's called a diploid or a 2N cell. When that DNA is replicated, and we've now doubled the amount of DNA in each chromosome, that means we'll still have 46 chromosomes. 46 stays the same, but each chromosome will have twice as much DNA as it did before. And we can see that represented by the fact that each chromosome will now have two strands. Each strand is called a chromatid. So now each chromosome is made up of two chromatids. The two chromatids are held together at the centromere. So we have 46 chromosomes, but now we have 46 double-stranded chromosomes. So we can call that a 4N cell, a 4N cell. And then that cell will continue through that first meiotic division. And what happens in that first meiotic division is that the pairs of chromosomes line up with one another. And then when this nucleus divides, one chromosome from each pair goes into each nucleus. So we'll have one chromosome from each pair in each nucleus. Remember, that still is a doubled chromosome, two chromatids in that chromosome. So now we'll have a nucleus that has only 23 chromosomes, one from each pair, but 23 double-stranded chromosomes, each with two chromatids. So we would call that a 2N cell again. Here we had a 46 chromosome 2N cell. Now we have a 23 chromosome 2N cell. Half as many chromosomes, but twice as much DNA in each chromosome. The two cells that contain those two nuclei will be called secondary spermatocytes or secondary oocytes. And those cells, secondary spermatocytes or secondary oocytes, will then enter the second meiotic division, the second meiotic division. And what happens in the second meiotic division is that in each chromosome, the centromere splits, allowing the chromatids to separate from one another. And when the nucleus divides, one chromatid enters each nucleus. And so now we'll only have a single-stranded chromosome. So now we stop calling it a chromatid. We just call it a chromosome again. We'll only call it a chromatid if there are two of them for each chromosome. Now we'll just call it a chromosome. So each chromosome now has half as much DNA as it did before. And each nucleus now has 23 chromosomes, just like it did here, but only one strand, so half as much DNA. So that would be a 23 chromosome 1N cell or a haploid cell. And those cells, the cells containing those nuclei, are the definitive gametes, either the sperm or the ova. Okay? So that is what's happening in the nucleus, regardless of whether it's male or female. First meiotic division, giving us secondary spermatocytes or secondary oocytes, and then the second meiotic division giving us the gametes. Okay, Now, what is different in males and females? And as I said, what's going to be different is what's happening in the cytoplasm of the cell. So now let's have the circles represent the entire cell, not just the nucleus, and see what happens. What we're looking at on this diagram fundamentally would represent what happens in males. We start off with one primary spermatocyte. When that divides, we end up with two cells, each one having its own nucleus, and those are the secondary spermatocytes. And when they divide, we end up with four cells. Those are the spermatids. Okay? So we end up with four sperm developing from a single primary spermatocyte. That's what happens in males. In other words, each time the nucleus divides, the cytoplasm divides, it gives us two cells. In females, something quite different happens. In females, when the primary oocyte divides at the end of the first meiotic division, we end up with the two nuclei. We've already said that. But when the cytoplasm divides, instead of the cytoplasm dividing equally between the two cells, almost all the cytoplasm stays with one nucleus. Almost all the cytoplasm stays with one nucleus, and very little cytoplasm, just a thin rim of cytoplasm, stays with the other nucleus. The cell that contains most of the cytoplasm is the cell that we will call the secondary oocyte. The cell that has very little cytoplasm will be called the polar body. And the polar body is going to die. So this cell will die. So if this cell dies, then none of this can happen. So in the female, we would have something like that. We would have just a single secondary oocyte that will enter the second meiotic division. During the second meiotic division, we already know what happens in the nucleus. When the cytoplasm divides, again, almost all of the cytoplasm stays with one nucleus, and just a very thin rim of cytoplasm stays with the other nucleus. Again, the cell that has very little cytoplasm will be called the polar body. You can call it the second polar body, and that will die, leaving us just one cell, and that's the cell that we call the ovum. So what we've accomplished by doing this, by dividing the cytoplasm unequally so that almost all the cytoplasm stays with just one of the cells, with very little going to the other, is we're conserving all of those cytoplasmic organelles, like the mitochondria, the ribosomes, the endoplasmic reticulum, all of those organelles that are going to be necessary for energy production and protein synthesis, that's going to be needed if the cell is fertilized. So rather than splitting all these organelles between multiple cells and thereby wasting all of those organelles in cells that will never get fertilized, we conserve them and keep them all in the one cell that might be fertilized, the cell that becomes the ovum. Okay? So that's what happens in females. Okay? So in females, in contrast to males, out of a single primary oocyte, we end up with only one ovum. Remember, in males, out of a single primary spermatocyte, we ended up with 4 sperm. Okay, let's go back to our original diagram and talk about the timing of these events. The timing of these events. First, in males. In males, the process of differentiation to go from a mitotically active spermatogonium to a meiotically active primary spermatocyte, that process does not start until after puberty. In other words, before puberty, in the male testis before puberty, the only germ cells we have are spermatogonia. In other words, they are somatic cells, and they are dividing by mitosis. So there is no meiotic activity at all in the testis before puberty. We just have spermatogonia dividing and giving us more and more spermatogonia by mitosis. Then after puberty, then the differentiation process begins, and those spermatogonia start differentiating into primary spermatocytes, which are meiotically active, which then go through the 2 cell divisions of meiosis to give us the sperm. That process of meiosis then is going on continuously, day and night, 7 days a week, from puberty until death. A continuous meiotic process continually giving us spermatids. Okay? So in the male, none of this is happening before puberty, and then it's happening continuously after puberty. Okay? That's in males. In females, it's very different. In females, the process of differentiation from oogonia to primary oocytes begins well before birth. Starting at around the 5th month of development, we start to see differentiation of oogonia into primary oocytes. By the time this female reaches 9 months, by the time the baby is born, all of the oogonia have either differentiated into primary oocytes or have died. So in other words, in the newborn female, there are no oogonia and there is no mitosis of oogonia because all of the oogonia have either died or have differentiated into primary oocytes. Now remember, if they are primary oocytes, by definition, that means they are meiotically active. So in fact, the process of meiosis begins before birth. And these primary oocytes start their first meiotic division. But they don't get very far. They don't complete that first meiotic division. They only get partway into it. They get as far as prophase of the first meiotic division, and then the process stops. And that's where these cells remain, from before birth until puberty. So in the ovary of a prepubescent female, what we have is lots of primary oocytes arrested in prophase of the first meiotic division. And they stay arrested until after puberty. Now, each month after puberty, usually one, sometimes it could be more than one, but usually one of those primary oocytes will pick up the process from where it left off. It had stopped in prophase. So one of those primary oocytes will continue that first meiotic division, complete the first meiotic division, give off a polar body, give us a secondary oocyte. That secondary oocyte will start the second meiotic division. It will get partway into that second meiotic division. This time it will get as far as metaphase of the second meiotic division, and then it stops again. And it's at that stage, at metaphase of the second meiotic division, when it will be ovulated. So what is ovulated each month is a secondary oocyte arrested in metaphase. Now keep in mind that that secondary oocyte that's arrested in metaphase had been arrested in prophase of the first meiotic division as a primary oocyte and had been arrested for many, many years, depending upon how old the woman is at that particular menstrual cycle. It may have been arrested for 20 years, 30 years, 40 years, before finally getting through the process to ovulate as a secondary oocyte arrested at metaphase. So what is ovulated each month is not an ovum, but is a secondary oocyte arrested in metaphase. Now that secondary oocyte will never finish that second meiotic division unless it is fertilized. If it is not fertilized, that's as far as it gets, metaphase of the second meiotic division, and it dies as a secondary oocyte. But if it is fertilized, then one of the things that will occur after fertilization is that that second meiotic division will complete, will pick up from where it left off at metaphase, will finish the second meiotic division, give off the second polar body, give us an ovum. But remember, that ovum already has a sperm in it, right? Because it was fertilized. That's why it finished the second meiotic division. So it is a fertilized ovum. And then the nucleus of that ovum and the nucleus of the sperm will fuse, and now we'll have a zygote. And we will have now restored the 46 chromosomes, 23 chromosomes from the ovum and 23 chromosomes from the sperm. Okay, so that will give us a 46 chromosome diploid cell. Okay, so that's what happens in the female.